My name is Noam Shoresh. I lead the computational arm of the Epigenomics production group here at the BroadE. Some members of our group are here. But I'll try to talk to you about, or describe to you a little bit of what the type of data that Chuck spent time explaining more detail, how you acquire it and what's its nature, how that could be used to help us understand some interesting things. But I think it's really flexible. We don't know exactly who you are, what you know, what you hope to get from this. So if you want to take any direction that's not quite the main line of my slides, feel free to pull us in that direction. And what I can't answer, Chuck or Alon probably can when Alon resurfaces. So, okay. Part one I'm going to describe to you is a condensation of the work of a lot of people, and in particular the program and the Bernstein lab and Analyses lab and other people, and ENCODE and different groups in ENCODE. So a true acknowledgement slide would be too large. So very basic, and one way to ask a lot of questions in biology is we really try to understand what does this thing do, what does the genome do? And what do we know about the genome? So now we know the sequence. We can look at the sequence. And the sequence tells us a lot. If we just look at the sequence, we can find patterns here. And if we compare different species, we can, and the sequences of different species, we can find more patterns. And for example, one thing you can learn just by looking at the sequence is where genes are. So some parts here have a pretty clear sequence signature that's telling you there's a gene over there. Some part there that's coded for a protein. Okay, how does something like that help us? For example, there is this big field of genome-wide association studies. That's a type of studies where you have, let's say, a disease, and you conduct a very large survey with thousands, sometimes many thousands of people, and some of them have that disease and some don't have that disease, and you look at their genomes. And you're looking for mutations that are of higher frequency within the group of people that have the disease, and these are very subtle signals, and it's not easy to pick them up rigorously without lots of false positives, but it can be done, it has been done. So from these type of studies, what you come out with is a list of hits. They're called SNPs, single nucleotide polymorphisms, mutations, some places in the genome where if you have this letter instead of that letter, your chances of having, let's say, a disease is somewhat higher, and it's been shown significantly, to be statistically significant. So when you look at these locations that are found by these studies are completely blind to what's underneath the sequence. They just look at the sequence itself. So maybe you find a mutation here that's associated with a disease, and you say, okay, I have some understanding from this, because it occurred in part of the sequence that is coding for a protein. So when I had this person with that mutation, this gene is now going to produce a different type of protein that with an altered function, and you can start following up the consequences from that. But quite a large part, I think more than half of the mutations that are found to be associated with diseases fall somewhere here, and we don't know much about them. And what I would like to describe to you in part today is how the type of data that you acquire with ChIP-Seq helps annotate big parts of the genome that until now were not annotated, so that we do have a better idea of what a mutation somewhere here may be doing. So I'm going to talk about epigenomics, but I want to start with a little bit of a distinction that I learned shortly after I joined the group, and I think it's a fun one. It's something to impress your friends with. All the cells in our bodies have the same DNA sequence, right? And yet, a liver cell, when it divides, it doesn't suddenly become a muscle cell. So there is some information that is not the DNA itself, it's not the genome itself, that's transferred from a liver cell to the daughter liver cell that makes it stay a liver cell. And any type of information like that that is hereditary and is not the sequence itself is called epigenetic, okay? So that falls under epigenetics. Epigenetics. So this is and beyond the sequence. And the epi here refers to this beyond the sequence. Well, it doesn't say epigenetics up there, it says epigenomics. And this is slightly different, there is an overlap. But the idea is somewhere in the cell is this information stored that is epigenetic information. And one of the likely places it is stored is on the genome itself. So the DNA is here, and there are all kinds of things coming and binding here and modifying here. And we heard about the histone modifications. So the DNA is wrapped around these beads, these histone complexes, and they have tails. And it turns out that modifications of these tails, chemical modifications of these tails, can change how tightly or loosely DNA is wrapped around the histone, for example. And that can affect how easily some genes are transcribed or not. So there is a lot of information that is associated with the genometry on the genome at various locations. So epigenomics in a way much simpler concept, and that is literally on top of the genome. So anything that's associated with particular locations on the genome, but are not necessarily, so that could be a protein that's bound somewhere or a histone modification. So what we really study with the ChIP-Seq methods, right, we have some part of the process, I'm going to go over it very quickly again, and Chuck already talked about it, that has to do with finding parts of the, enriching the part of the, so finding the parts of the sequence that carry some molecule that we care about, and then figuring out what these bits are. And that's what we do in our production group, with a lot of thought about this. Whatever we find that is epigenomic is part of epigenetics as well. It's implicated in differentiation. I'm not implicated. It's part of the differentiation process, and so on. So just a bit of that. All kinds of things can happen. I just mentioned a few. You can have proteins that are bound, transcription factors. You can have. You can talk about, when you talk about epigenomic features, you can talk about DNA methylation. I'm gonna, in a way, talk about histone modification. In a way, not much that I will say will be too specific to histone, because I'm not going to be. Not much that I'm going to say is going to be too specific to anything. It's more some broad concepts. Okay, so kind of recap of Chuck's explanation. He gave you a closer look at some details that are important to you when you're trying to understand the data sometime, and you need to figure out, or if you're planning to do an experiment like this, is going to be the bird's eye view of the same process. What is ChIP-Seq? There are two parts. So there is, we want to find where on the genome is a certain target, something that's interesting to us. Maybe a histone modification, maybe a transcription factor. We start by breaking the genome to small bits, and we introduce antibodies that are designed to mostly bind to our target. Okay, they don't always do just that, but mostly that. At this point, we can clean everything that doesn't have the antibody. In other words, this is the pull down. We pull only the parts that contain the antibody. Once we did that, we don't need the antibody anymore. In fact, we don't need anything but the DNA sequences, because we're going to use the sequences themselves. We sequence them, And we use the information from the sequencing to map them back to, because we know the reference genome. If you want, I can spend a little bit of time about this and related to the question that was asked before, single end, 36, 100, paired end. I mean, what is, I can say one sentence and then depending on if you want or not, we can, I can say a little bit more, but we do 36 base sequencing. Single end, what does that mean? These are DNA fragments that came from the assay, but use the board. Right, this is a fragment. This is a piece of DNA fragment. It can be 200, 300, depending on how it's not completely uniform. 200 base pairs, long set. When you sequence it, you have a choice. In the Illumina, you prepare the library and how you run the machine. You can read the sequences from just one side or from both sides, and you can read this far or more, right? You have these choices. When people, and that was a lot at the kind of beginning of the sequencing applications and uses, and it's still being done, right? If you want to sequence some new organism, de novo, you don't know what you're doing, you're trying to get large chunks as much as you can. That makes putting back the puzzle a lot easier. So if you can do 300, 400 long base pairs, if you can do paired end, assembling the original genome is much, much easier. But for a lot of the something-seq technologies, all you really want in the end is enough sequence to be able to map things back to where they are in the reference genome. So 36 bases is pretty good to recover, I think, 80% of the genome, something like that. Okay? What does that mean? Why wouldn't you be able to recover everything? Some parts of the genome are low complexity, so the nucleotide composition there is very simple, and it's replicated. There are many, so there are regions of the genome. Let's look at the simplest example. If you have you know, a few hundred times, if you had something like that, and if you're you sequenced a bit of this and you got 36 A's, well, where do you align it to? Here, 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 here. This is a very simplified example, oversimplified example, but situations like this do occur a lot. There are parts of the genomes that are low complexity, that are, so when you have a map of even 36 bases, you can't map it back uniquely to where it came from. And I'll say no more unless you want me to say more on this. I think this is a no. Okay. So this is what you get in the end, right? You pile up the reads and you get these maps. These are chromatin maps that we look at, we know and love, and so what? What do you get from this? So, part of the ENCODE project, ENCODE is a large consortium now entering its kind of third phase, and as part of that, so I'm sorry, its acronym is Encyclopedia of DNA Elements. So the idea is, the goal is that we would say something about every bit of sequence here that we can, to say something about it. What is it doing? What's it important for? And as part of that, our group and some others did a lot of ChIP-Seq assaying and creating maps for many different cell types, many different histone modifications. And I'm going to talk a little bit about one kind of pioneering study that came out of Analyses and kind of BroadE's groups. So we're talking about 10 modifications, about 10 cell types. And the first thing that emerges when you look at it is that, sure, there is something going on under genes, but there is a lot of non-random stuff going elsewhere. It's not just genes is where everything makes sense and everything out there is just randomly distributed. It's not like that at all. There is a lot of non-random stuff going on. You find in particular, you find certain combinations. There are many, many ways you can combine 10 different things. But not all of them occur. You don't always find out, you don't find if I told you, okay, pick 5 of them, do you find a lot of places in the genome that have this, and this? The answer is no. There is a subset of combinations of histone modifications that seem to be common. Those are the ones that probably do something, and they occur, and we could identify what are these combinations. It ranges between, you can decide a little bit on how fine resolution you want. You could think about it as 10 combinations or 30 combinations. It doesn't matter compared to the, how many are there? 2 to the 10, there's a large number. All the combinations you could have. There are, so in this work, I think we settled on, or they settled on 15 states, but these are combinations of histone modifications that seem to be recurring a lot in many different cell types, and a lot in the genome, and they were associated with things that make sense. So one combination seemed to have to be marking genes, the promoter of genes that are active. And another thing seemed to be marking genes that were suppressed. And actually, Chuck showed you a couple of these. That's from 2007 from Target, but this work has kind of rediscovered that and more. In particular, there were things were found that are more interesting in the sense that they are now, because we can tell about something that's coding not just by the combination of histone modifications. There are assays today, right? You can do RNA-seq and you can figure out, oh yeah, this part of the sequence gets transcribed. But for all these things, there isn't much orthogonal ways of getting at what's going on here. And some of these distal elements seem to be, there was particular interest in enhancers. So these would be genomic elements, some DNA bits of sequence that carry a particular set of marks, set of histone modifications when they're active, and not have those when they're not active. And when they're active, they're thought to be remote switches to some other gene or genes. Okay, the mechanism is not quite known. A lot is, there are conjectures and some evidence about 3D looping. So the enhancer that seems to be far away from the gene actually loops back and is very close to the promoter. But it needs that chromatin structure around it, those histone modification complex or combination of histone modifications to be active and to start. These are enhancers. So in that work, kind of showed the way for a lot of similar projects that are underway right now and that followed after this. And the concepts are very general and I think very interesting. So let me explain. So you can find, using this data, you say, okay, now I know where I'm going to have cell type 1, cell type 2, let's start with cell type 1, okay? I can list here all kinds of enhancers that I find that seem to be active in cell type 1. So these would be places in the genome that show this combination of histones that mark an enhancer, an active enhancer. And I say, okay, that's active here, and this one is active here. And actually, we do this for several cell types, and we combine them, so we can get this kind of master list of all the enhancer elements in the genome that we find when we ask, let's say, 10 or 15 cell types. And we can start, for each element, we could say, oh, this element is active in cell type 1 and 2, this one only in 2, this one only in 5. So we can start getting this, this annotated list of enhancers, and a particular enhancer, we would know in what cell types it's active or not. That's interesting information. Oh, here it is actually. This is the kind of list I was talking about. So this is from work done in our team by Sam Lee. And so she did that. She processed about 100 different cell types that we have data for which we have the histone modifications that are kind of relevant for enhancers are, if it means anything to you, H3K27 acetylation, K4MB1, and K4MB3. That's what we're basing our calls on. People are still debating what's the best way to call this, and there are definitely things to consider, but this is a pretty good approximation. And then you combine them all into one single list of all the candidate enhancers. So that's what you're seeing here. It's kind of, this is not just running genomic coordinates. These are separate regions you see. So each column here is a single element. The fact that it's black here means it is a candidate enhancer. So it's an enhancer that was found to be active in some cell type. And below you can see which cell types it was found to be on. If you see something here that doesn't have any green under it, that's because the list that was used to produce that is 100 cell types long, and I'm only showing you here 5 or 6. And an interesting thing, for example, to see here, so here we're not just looking at these enhancers. So I'm now going back to that question of the GWAS, right? We said that GWAS SNPs, you remember those mutations that are associated with disease sometimes fall far away from genes, and we don't know what they do. So this particular list that you're looking at was further filtered down to be just around SNPs. So in fact, up here you don't have all this information. It's not really important, but each column here is actually a little region around a mutation, a SNP that was found significant in its association with a certain disease. And this is all the GWAS studies combined, so it's many diseases are shown on this track, okay? And then we filter down from all those, only those that occurred in enhancers. And you see that quite a few of them do fall in enhancers. That's already interesting. And if you look at the cell types here, we have some blood cell types here. This is an embryonic stem cell. I don't even remember what all the others are, but here is brain. And you can see, for example, here a collection of enhancers that seem to be, so these enhancers all seem to be specific to the brain. They're active in the brain, but not in other cell types. And when you ask, okay, they each appear in a different region here, and they're associated with some SNP, okay? So, and each SNP comes from a study, and you can ask, okay, what disease is associated with these mutations? It turns out most of the things that you find here are Alzheimer's. That's kind of rewarding. You have actually created a connection, a potential mechanistic connection between a mutation that is associated with a disease and a regulatory element, something that can turn genes on and off and seems to be active just in the brain. So it makes sense that if something is not functioning well, if there's a mutation that's disrupting the function of this element, it may lead to phenotypes that are associated with brain function. So to do this kind of analysis, what you really want to do once you identify all the enhancer elements in the different cell types, you want to group them. You want to find sets of enhancers that seem to have similar profiles across cell types, because they have some associated function. They seem to be on in associated contexts. So it makes sense that whatever turns them on may have a certain signature. So you collect all the enhancers that show a similar profile. So let's say all the enhancers that are on just in brain or just in a particular cell type, and you look for motifs there. And if you find enriched sequence motifs there, then maybe you have found the trigger, the thing that guides maybe a certain transcription factor to all these enhancers in a particular context and turns them on. I don't know if I have the... okay, let me explain that a little bit more. Right, so we group the enhancers from that picture I showed you before. You group them into a set of them that are on in a particular cell type, okay? So here we have enhancer group 1, say. So these are all these are different cell types, and you find a group that is on mostly here and maybe here, and it's kind of on here. And you look at this, this may be, I mean, we're talking about maybe 500,000 enhancers genome-wide from these studies. And then when I talk about finding groups of hundreds of enhancers, or thousands at most, I think hundreds. And when you look for motifs, you find that there is some motif enriched under these sequences. Okay, in these elements, there is a motif. And you can then even ask, okay, is it a known motif? Is it a motif that I know what transcription factor used that motif as a binding signal? And you find, yes, there is a certain transcription factor, right? So you find from this, you find motif, and you find a transcription factor associated with that motif. And then you can ask, okay, when is this transcription factor expressed? And what do you expect? When would you, what kind of profile of expression of the transcription factor would make sense with the story we want to tell about this enhancer? It's a question to the audience. Okay, if everybody's with me, which may not be the case. We have a set of switches here that seem to be on in two cell types. And they seem to contain a unit within them that is something that a particular transcription factor can go and bind to. So now we ask at that transcription factor, that molecule, and we ask, okay, when is this expressed in the cell? So what if we find, well, what do you think we find, because the way I set it up?
It should be expressed, right?
It should be expressed in the same cell types that this is on. And I can't say that it's expressed exactly here and here, but there has definitely been found strong correlations between the profile of expression of certain transcription factors and the profile of activity of the enhancers in particular enhancer sets. So,
just for a little more background. So
sure.
How is an enhancer defined as active? If it's not the motif, is this the modifications?
It is the motif.
Functional.
It is a, in a way I should say, putatively active. It is defined by the histomodifications. So if we find the signal of K4ME3, K4ME1, K27 acetylation in some combination, we say that's active. What we really mean by that is we're guessing it's active. In a few handful of cases that we could test it functionally, so we can put it in those few cases, it bears out sometimes. A big effort in the field now is coming up with high throughput ways of functionally testing these conjectures. But this first started because this combination of histomodifications seem to be associated with P300, which is another thing that traditionally been thought to be associated with active enhancers. But it's really a name we give to a particular combination of histomarks that we think is that. Yes?
But you can actually do chip on that transcription factor, right? You can see whether they bind.
We could. Well, for some of them you could, yes. Yes. I don't know. For this data set, that hasn't been done, I think, for the transcription. Yes?
I think it's a comparison myself.
Sorry?
The word motif means nothing to me, sorry.
I'm sorry. I'm really sorry.
No, it's okay. Yeah, no, if you could just put like a sentence about what that.
I will backtrack.
Okay.
I'm very sorry about this. So the question was about motif. So I told you we find bits of DNA that seem to have associated with them certain patterns of histomodifications. Okay, so I found a pattern and I call that active enhancer. Okay, but that's just a name. It's really in this part of the genome. I find that certain events happen that I measure with these maps. Okay. In particular, when I look across cell types, I can group them. So, these lines indicate parts of the DNA sequence that marks an enhancer motif, and now I'm collecting together a bunch of them, maybe 100 of them, that seem to be on, so they seem to be active in the same cell types, okay? Now I look under the hood. I look at the sequences, because each of these may be 1 to 2 kilobases, 2,000 base pairs of sequence. A, G, T, 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 C, da, 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 the same. Motif is short for sequence motif, is a combination of 6 letters, 8 letters, something like that. And
it's like the columns in the exochip figure.
It is a combination of letters that satisfy several conditions, but in a way it's thought to be what, if there is a protein that interacts with DNA, it needs to recognize some part of the sequence to know this is where I need to go. So a sequence motif is just that. It can be a 6 to, or whatever, A, G, C, usually it's, I don't know if you've seen things like that are written C, A, E. They have some way of writing them depending on the probabilities you expect, but this is an unnecessary complication. Okay, it can be something like this, and it's something that, I don't know historically or previously how things like this have been found, but you find, oh, a particular transcription factor that I know about seems to bind to this sequence. So there's some information known about that. Now, if you take some set of sequences, especially if you take 2 larger sequences, let's say all the enhancers, and you ask, statistically speaking, do I find any 6-letter or 8-letter sequence overrepresented or surprisingly represented? The answer is no. If you have noise, you will not be able to find it. But once you kind of trim it down to a set of enhancers that seem to have common profile of activity across cell types, and you ask, okay, among all these sequences, does something come up? So you can run a program that scans all these thousands of sequences and tell you, yes, you know, in this set, A, G, G, C, T, A, G, and A, C, A, C, da, 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 these 2 are statistically significantly appear more in this sequence than in kind of a null model where you just look at random sequences. And that makes you think, oh, okay, so something may be using these sequences to turn these enhancers on. And that thing, sometimes you have additional information that's telling you, oh, I know which transcription factor, which are transcription factors always proteins, I guess? Which protein binds here, okay? So I can now go and say, well, but if this is the thing that activates these elements, and these elements are only on in the brain, I'd better find that this transcription factor is on in the brain as well, because if it's not, then my hypothesis doesn't make much sense. And it turns out it is. There is correlation between the sequences that were found under this set of enhancers and the expression of the transcription factor associated with these motifs. Okay?
So my question is,
Please.
so you're grouping them based on activity in a cell. So let's say you find one group, let's say brain selected group, and another is lung selected group. So within that brain selected group, are you subgrouping them again based on their histone modification status? Or, I mean, I would expect to see a diversity of enhancers, because in the brain cells, you don't have to have just one type of enhancer coming up because of one type of transcription factor. So you're gonna get, like, I don't know, hundreds of enhancer types. So I'm gonna like subgroup them based on all the different. So I don't know how that works.
So the question is about subgrouping. Let's say you found enhancers that are active in the brain, but that's not really one thing. There are different things that are going on just in the brain, and is there a kind of subcategorization? I think the first pass is to find these sets that, by looking at the cell-dependent profile. You don't distinguish them by the histone state, because that's what you started with to begin with. All of these had more or less the same histone state, meaning they are active enhancers. So they show whatever, K27 acetylation and some degree of K4ME1 and little, okay? So they had.
So you initially self-classified them based on the whatever store box, and then that's what you put into your analysis.
This is the element we call enhancer. You could do a similar thing with active promoters. Yes. Oh, mine stopped working.
Recording. Well, no.
Oh, I've been using the wrong one the whole time?
Yeah.
I'm sorry about that.
That's okay, thank you.
This is absolutely. They could hear me anyway. You could do the same analysis for a different element type. Let's say all the active promoters that are specific to the brain. You find less of them. It turns out promoters are slightly less dynamic. So when they're on in one cell type, they will be kind of on in several related cell types. But that doesn't mean that enhancers shouldn't subclassify as well. When you do these, you find enrichments. But when you go back and ask, of these 1,000 enhancers, how many of them actually have this motif? You'll find some percent of them, maybe 20%, maybe 40%. So there is more going on there definitely to kind of take apart and find more high resolution. Sure. Any more questions? Yes?
To identify a motif reliably, like how much coverage do you need at that region?
To identify a...
Because there is a background noise as well, right, when you compare. So you need like 30x, 20x, that's enough to...
Oh, we're not doing...
Because you're aligning the reads, right?
We're aligning the reads to this region,
right?
Okay. So we find in this region, if we put some, what I said, K27 acetylation, K4MY, K4, we find that there's a peak here and some other peak like that and something like that. You say, okay, this is what we call the enhancer element. So that would be 2 KB. Okay. Now the sequence of this we don't get from de novo sequencing. We use the reference genome.
Oh, okay.
Okay. So we use the reference genome, and but more details about what it takes. I mean, I don't have myself a lot of experience with playing this game. So like, how much noise would drown you? What happens if you include too much or too little? What's going to? One interesting thing though is the motifs seem to occur in places that are slightly more conserved in evolution, which is not very surprising. And they also correspond to dips in the histone map. So and you would find, even in the K27, basically the place where the transcription factor binds, it leaves room, so it's pushing the nucleosomes aside. So there is a dip in the signal in all of these tracks, and a peak in another assay that's kind of testing for how open DNA is. And so there is this big interest in kind of, this is probably where the motif is. And in these dips, and they're considered a good way to hone in exactly within that element, what is the part where you have the binding and the activities going on. Okay? How am I doing with time, by the way? What is 2 o'clock? Okay, I'll wrap up. Right, so for each of these new elements, there is a wealth of information now that we add. Unlike a gene is a gene, it's there. But yeah, it's interesting to know when a gene is on and off. Now we can tell about all these, for example, enhancers, we can say the same thing. What are the cells in which they're active? What are the important subsequences? So that's kind of the motifs and upstream, what turns them on and off. There's a big interest in, okay, you found an enhancer. Which gene is it actually regulating? Which gene, when this enhancer is active, what gene is on because of that? That's a non-trivial question. There are all kinds of attempts to answer that. It's kind of just emerging. Sometimes it's a nearby gene. Very often, the answer is not so obvious, and you can look at correlations, and there are a lot of attempts at how to address that, but that's definitely a direction, but that's going downstream, right? What is it regulating? And this is kind of an example of this type of, the success of this approach. And when I made this slide several months ago, I remembered the details. I must say, I don't right now, but I think these are 2 examples of GWAS SNPs. So this is an example here that was found to be, that previously was not known what it was doing, but it turned out so it's associated with leukemia. Is that right? No. I'm sorry. I don't remember what it's associated with, but I think the point is you find that it's hitting an enhancer that is active in blood cell type, and it made sense for the disease it was associated with that I can't remember what it was. Moreover, if you look even more closely, you see that it's disrupting a motif that was found under that enhancer. So you have an even better functional understanding of what's going on. It's disrupting a motif. So transcription factor that was supposed to turn this enhancer on cannot do that anymore because the motif is disrupted. And there is information in this case about what gene this enhancer regulates, and it turns out to be a gene already implicated in the same disease. So we have a pretty impressively mechanistic story, full story about a mutation that is associated with a disease that opens up the arena for all kinds of treatment strategies, drug development strategies that you couldn't do before. I'm sorry?
I think it says that it's
lupus. But are both of them? This one is leukemia, that one's lupus. So, yeah, I think that's it. If you have more questions, I'd be happy to take them on anything I talked about or other. And if not, I think we can take, go ahead.
So, the enhancer, I know, okay, this will be right. If it is wrong, it's wrong. Which one is
So the question is, so that the enhancer elements themselves are relatively large. They can be, they typically are of the order of 2 kb, but yeah, they can be 5 kb. When we say that, we mean that's the region where we see the footprint of the histone modification. But under that, there are many motifs in principle. After this analysis, where you're looking at, and the question is which of these motifs is the one that kind of is the important one. First of all, maybe more than one. Maybe several motifs are relevant for a given enhancer. But really, you don't just look at one of them all by itself. You look at a combination of, maybe 1,000 such enhancers. And then you find this sequence to emerge as significantly enriched. So now, even though there are several motifs in general, several things that in other situations have some meaning, under your enhancer, probably only one of these is found. One of those that are enriched in this set. So
Very cool. So the reason why, I don't know.
how many histone modifications define an enhancer?
Or combinations of modifications. Are there 7 flavors, 12 combinations?
I think in terms of the number, it really depends on, there is some understanding about the important ones being K4ME1 and K27 acetylation, so that's 2. But also K4ME2 seems to be important, so there is kind of, I don't think there is a set answer how many. There is between 2 and 5 is probably enough to determine what's that something is an enhancer. And there are several flavors. So in this original, in this work, there was a distinction between weak enhancer and strong enhancer, which had to do with, you find, I think it was the K27 acetylation that was the distinction. So they had K4ME1 and a little bit of K27 acetylation, it was weak. If they had K4ME1 and a lot of K27 acetylation, it was strong. There's also a little bit of echo between enhancers and promoters, because they are probably, at least in some cases, nearby physically. And when you cross-link and you pull with your antibody some chunk of some histone with whatever DNA is dangling there, you may be pulling an enhancer when you were actually targeting something at the promoter and vice versa. So there is some of the things that are important for promoters, like K4ME3, are also present in the enhancer, maybe because they are really there, and maybe because it's an echo. But there are between 3 and 5 probably important marks, and I wouldn't be surprised if in the coming years people will realize that there are a couple more marks that help make very good distinction between two classes of enhancers. Yes?
Yep. What is the connection, if any, between these kind of things and DNase hypersensitivity?
The connection between these things and DNase hypersensitivity. There is a lot of connection. So in the ENCODE, there were a couple of groups, most notably in Seattle, John Stem, the University of Washington, that assayed DNase hypersensitivity. So what does that mean? You digest the DNA, and there are some, with a particular enzyme, I don't know the details, I'm sure Chuck can explain a lot better than I do, but you get much higher probability of having the DNA cut in particular locations. So these locations are called DNase hypersensitive sites. And when you do that, you find many of these regions, many of these hotspots of DNase hypersensitive sites. And when you combine it with histomodification data, you find that they all often align exactly in the dip here, at the center of enhancers. And they did other things where they kind of compared the set of their findings with GWAS SNP database, and showed that a lot more SNPs, there is enrichment. If you're looking, instead of at the entire genome, you look at the set of locations right near DNase hypersensitive sites, you're more likely to find GWAS SNPs there, so mutations that are relevant for some function. And so some people treat that as the DNase hypersensitive sites as a way of calling kind of candidate enhancers, and it's probably a little, it's casting a slightly too wide a net, but I think what's emerging in the field is some combination of histones and DNase will allow you to pinpoint enhancers and perhaps exactly the binding location and stuff like that, but it's very closely related.